All right, Sarah. Hi. Thanks for joining.
Thanks so much for having me. It's so great to see you.
It's really great to see you too, and nice to see you in the office. What a strange and wonderful thing.
I know, yes, it is very, very bizarre. It's mostly empty here in the office, but the lab personnel are still reporting in and doing our thing. So
That's great and lucky, and hopefully more of us will be able to get back to normal soon. So maybe we can start just by having you introduce who you are and where you work and what you do.
sure, of course. Um, my name is Sarah Seaton, and I'm a senior scientist here in the Boston-based Indigo Agriculture, which is, as you might imagine from the name, it's an agricultural biotech startup that has a microbial connection in that our products in part are driven by natural microbes that we put on seeds to promote plant growth under stressful conditions. So we're really trying to promote a natural solution to some of the agricultural issues that are facing the planet, such as increased fertilizer use, decreased availability of water in many cases. And so we believe the microbiome of the plants can impart some sort of advantage to those struggling plants under stressful conditions.
It's so smart. It really feels so much like the future that we need to get towards, right? Where just a better relationship with biology and especially our food and agriculture. So what a wonderful job. And so you're a senior scientist? Are you in the lab most days, or are you running a group?
So that's a great question. Things have changed a little bit with sort of the COVID situation where we're trying to limit the number of people who are in the labs at any given time. So that means over the last year or so, I've actually got to spend a bit more time in the lab, but I do lead a team here of microbiologists and molecular biologists for what's called our microbiology and molecular core. So I have a team of about 6 currently, and we're in the labs mostly, and we're essentially isolating new microbes that go into our culture collection here. We now have tens of thousands of microbes housed in our freezers, because as you can imagine, for every one product that launches, thousands of microbes have to be tested and validated. So I have the coolest job, I think, of essentially running a microbe library here at Indigo.
That's such a great way to think about it, a microbe library. That's amazing. So, I think many of the BioBuilder students might be familiar with culturing some microbes like E. coli or maybe Saccharomyces cerevisiae, but culturing new microbes must have a whole new set of challenges to it, right?
Yeah, absolutely. So Indigo has kind of an interesting approach to what microbes we're sort of focusing on. So in the idea of sort of enriching for microorganisms that are best associated with plant health, what we do is we actually isolate directly from plant tissue. So we're focusing on what are called endophytes, and those are the microbes, the bacteria and fungi that physically live within plant tissue. So that actually adds another layer of exciting kinds of process to this, because my team is also responsible for sourcing those plants from the field. So in many cases, we get to actually go to the field, collect crop plants that are resilient to some environmental stress, bring those back to the lab, and then that serves as our source of the ultimate microbes. Mm. And to your point, a lot of these are more fastidious than your typical E. coli or yeast. So we isolate all sorts of really interesting microbes, many of which are novel species that haven't been described before. So as sort of an environmental microbiologist, you can imagine that being a microbe hunter for a job is pretty spectacular.
That is spectacular. Wow, a job that I'd be proud to have on my my calling card, right?
Definitely. Yeah, and we keep a little bin in the lab. We call it our show and tell bin for the organisms that look really dramatic on a plate. Either they're pigmented or some sort of really filamentous fungus that grows in some interesting way. So we keep those around for lab tours and to impress our non-microbiology friends who may not think of microbes as being really charismatic, but they absolutely can be.
It can be, yes, very colorful and diverse, and wow, what a great job. So, your background is in environmental microbiology, is that right?
Yes, yes. So my PhD is in microbiology. I got it from the University of Georgia. And my PhD projects were focused on taking environmental bacteria, soil bacteria, and modifying them to essentially degrade pollutants. So taking metabolic pathways, catabolic pathways in those bugs and improving their ability to actually degrade aromatic compounds that are often associated with things like oil spills or other contaminants in soil. So yeah, I've always really been interested in how we might harness bacteria to impact some environmental problem, and so that's where it all started.
That's so cool. And when you say always interested, so are you from Georgia? Is that did you grow up in an agricultural background, or I don't know how one, like I have no green thumb, so it would never occur to me to go in a plant.
Yeah, so I'm actually from North Carolina. I grew up there. All my family's still in North Carolina. I actually did grow up on a farm, on a tobacco farm. But I think I've always just been interested in sort of the power of microbes and how we might actually harness them to, solve different problems. Yeah, I think I knew I wanted to be a microbiologist when I was a freshman in college. So yeah, they just sort of struck me as interesting, and you peer through a microscope, and that's a memorable moment for a lot of students, I think, when you see these things for the first time. So and then being able, I think, to manipulate DNA. I won't tell you how long ago I was in grad school, maybe, but we've come a long way with being able to modify organisms, and then it was really exciting to be able to go in and put a new gene into a bacterium and see how that was actually able to completely alter the phenotype and allow that organism to do something it couldn't before you manipulated it. So yeah, that's sort of what really struck me as exciting. And at the time, it was much easier to do that in bacteria than it was in any other organism.
Right.
So that's sort of where I settled. Yeah.
Yeah, it's so great, the notion of harnessing the power of microorganisms to do useful things, and the control that we have, or some abilities that we have to manipulate the cells with DNA code. It's really at the heart of how BioBuilder teaches too. I think it comes to us as a surprise to students that biology is not just the study of the world as we've inherited it, right? But that you can add a layer of engineering to it to use biology to meet certain goals and endpoints. So you've been a wonderful mentor through our BioBuilder activities. Do you want to say a little bit about the students or about the interactions you've had?
Yeah, so yeah, we were very lucky 2 summers ago to actually host a BioBuilder student in the labs here at Indigo. So she was at the time a rising senior at Boston Latin School. Her name was Lynn. And yeah, she was such a breath of fresh air. She came in, and I think here at Indigo, things are really fast paced. I think that's probably the nature of most startups. So I can only imagine coming into that atmosphere, as a teenager. I'm sure that was very daunting, but she really just came in and contributed to the team almost immediately. So she isolated hundreds of microbes during her short time working in the labs. And yeah, I mean, she's the only student we've had as an intern here. I think because of COVID times, we haven't been able to host any interns for the last year, but I'm hopeful that that's something we can continue to support, because it was certainly like such a great experience for us, and hopefully for Lynn as well.
Oh, I know it was an amazing, really life-changing experience for Lynn. I think the opportunity for young, innovative, science and engineering interested students to have a chance to see science in action and engineering in action is really meaningful. It really can make it something that they have a little energy to pursue, right? They have a commitment to it once they've tasted the reality of working in that kind of a situation.
Right,
So
right.
yeah, thank you. Did you have somebody that sort of helped you along the way to commit to this career?
Oh, absolutely. There's been so many wonderful mentors along the way. I think I could list several, but I'll start with my PhD advisor, who actually I was lucky enough when I was in college. So I actually went to a small liberal arts school in North Carolina, which was a great teaching university, but not a lot of research going on a small campus like that. So I actually had the opportunity when I was still in my undergraduate days to do it's called an REU program, an NSF REU program. And I actually did that at the University of Georgia and met would later become my PhD advisor. So then she was able to recruit me to come back to Georgia for my PhD. And she's just always, even now, 15 years later, she still advocates for me, is always willing to listen if I have something that, I need advice on or need a recommendation letter. I think she's always really just been there and been supportive, both personally and professionally, which I think is really important when you're considering a mentor, someone you can rely on.
Well, I think she has clearly taught you well. What you touch on is so important. I also continue to rely on my network of mentors years and years after we've finished working together for personal questions, professional questions, letters that have to get written. We are all sort of turning around and helping the next one in line. So it is something that I think as a younger student, I didn't fully appreciate that these actually are lifelong professional interactions that you gain when you commit to working together on a project. So how wonderful.
Your network may grow as you advance in your career, but I think the really special relationships are the ones that you sort of maintain over the course of, several decades in some cases, which is
Absolutely.
really nice.
Well, I am sure that's what makes you such a wonderful mentor and that it made such a difference to Lynn as well. I sure do hope that we can get more BioBuilder students there working with you. Your work is fascinating and you're just a wonderful teacher and role model for them.
So thank you so much. That's very kind.